Hey everyone, and welcome. I am absolutely thrilled to have you here for this explainer. Today, we're doing something really cool. We are exploring the literal, physical architecture of living organisms. We're going to build our understanding from the tiniest structural building blocks and then zoom way out to look at specialized branches, specifically focusing on comparative anatomy. Let's get right to it.
Okay, let's dive into this. Form is closely related to function in all living things. Honestly, this is the golden rule of anatomy. The physical structure of an organism, its form, invariably dictates its physiological capabilities. So, it doesn't really matter if we're looking at a microscopic cell or a massive femur bone. The way a structure is built tells us exactly what it's designed to do.
Here's our roadmap for today. It's kind of like a zoom-in, zoom-out approach. First up, the science of structure. Then, we'll look at the macroscopic and microscopic worlds, followed by regional and systemic approaches. After we've got those basics down, we'll zoom all the way out through the comparative anatomy lens and wrap up by looking at skeletons across species.
All right, part 1, the science of structure, setting the foundation.
So, anatomy and physiology. They're distinct, sure, but they are deeply inextricable. Think of anatomy as mapping the physical structure itself, while physiology examines the active, life-supporting functions happening right there within that architecture. You simply cannot manipulate the physiology without first mastering the anatomy. They exist together as this perfectly interdependent matrix.
Moving on to part 2, macroscopic and microscopic worlds, a matter of scale.
And this brilliantly illustrates a major shift in perspective. Gross anatomy, which is also called macroscopic anatomy, covers the big stuff, the large body parts visible to the naked eye that historically were studied through physical dissection. Microscopic anatomy, on the other hand, dives into the unseen architecture. You literally need magnification devices to decode the structural logic at play down there.
Zooming into that unseen architecture gives us cytology, which is the study of individual cells, and histology, the study of tissues. These are highly specialized branches of anatomy. They allow scientists to isolate variables and observe the literal molecular building blocks of life. And grasping this microspecialization sets the perfect stage for when we do the exact opposite later on, scaling all the way out to compare entire species.
Which brings us to part 3, regional and systemic approaches, organizing the body.
You know, anatomists have to organize massive amounts of structural data, and they basically use two distinct methodologies to do it. Regional anatomy studies all the interconnected structures interacting in one specific area. Think of the abdomen, for example, analyzing how muscles, nerves, and blood vessels all pack together in that single space. Systemic anatomy, though, traces a single functional network throughout the entire body, completely regardless of location.
Looking at a few of the major organ systems here. Systemic anatomy reveals exactly how organs group together, like the heart and vessels working as a team in the cardiovascular system. The whole goal is to maintain human life and keep the body in a state of balance, which we call homeostasis. It's very much a network-level view of function.
Now, navigating all these complex regional and systemic branches requires a universal language. Standardized directional terms and planes ensure that no matter who is looking at a patient or how that patient is positioned, the scientific description remains perfectly accurate. For instance, describing the heart as medial to the lungs guarantees everyone knows it's located right toward the body's midline.
All right, part 4, the comparative anatomy lens, the ultimate zoom out.
Let's move to and see how this builds into comparative anatomy. This is the study of structural similarities and differences across different species. If cytology and histology are specialized branches that isolate the microscopic world, well, comparative anatomy is the specialized branch that scales back out. It leverages those exact same foundational principles to give us profound evolutionary context. It answers the ultimate question: why is the human machine structured the way it is?
Just think about the transition of animals from water to land. It meant dealing with the heavy pull of gravity and a sudden lack of buoyancy. Evolution had to shape physical structures step by step to combat those massive environmental pressures. It's actually why modern whales still have remnant pelvic bones buried deep in their tissue. Comparative anatomy lets us read those structural changes like an evolutionary diary.
And that leads right into part 5, skeletons across species, evolutionary evidence.
Now, what's really interesting about this comparative data is the massive structural contrast. On one side, we have the hydrostatic skeleton. You find this in soft-bodied creatures like sea anemones, and it relies entirely on fluid-filled compartments resisting compression. Contrast that with the human endoskeleton. That's a flexible, solid scaffold of internal bones and cartilage perfectly adapted to support massive weight against gravity. Two completely different structural architectures solving the exact same functional problem.
So, the crucial point here is that we don't just see these adaptive differences between vastly different species, we see them intraspecies as well. Take the human pelvis, for example. The male pelvis is typically a heavier and narrower structure. Meanwhile, the female pelvis is lighter, wider, shallower, and features a significantly broader pubic angle.
And this difference exists because, over generations, the human female pelvis adapted structurally to ensure reproductive fitness and successful childbirth. This ties right back into that core rule we established at the very beginning: form follows function. Every single biological reality, at every scale of observation, is shaped by what it needs to accomplish to survive. I want to leave you all with a provocative question to think about. Based on our drastically changing modern environment, how we sit at desks all day, stare at screens, and interact with technology, what future evolutionary adaptations might be studied by comparative anatomists centuries from now? How is our physical form going to change to match our new functions? It's pretty fascinating to think about. Thank you so much for your focus and curiosity today. Keep asking questions and keep exploring the incredible architecture of life.